Hello everyone. You guys never see me, right? So I'm Eugene again. I'm assistant chair again. You guys have to see me another time. For now, we have a lightning talk. It's gonna be 4 minutes, and then we're gonna have a 1 minute Q&A. So it's gonna be very short. I only gonna able to have one or two questions. So first person who presented is Frida Miranda. She's from the Oklahoma State University Center for Health Science, and also she have a poster outside, number 32. 32, okay? So the title is Characterizing Chimeric von Willebrand Factor in the ADMTS 13 interaction using Alto SPR.
Hi, everybody. Thank you for letting me talk today real quick. So first, a big broad picture overview about these two proteins and why they're important. So thrombotic thrombocytopenic purpura, it's a mouthful, so TTP for short, is a rare blood clotting disorder with an incidence of about 2 per million per year. So when there's injury to a blood vessel, the site recruits initial proteins to initiate primary hemostasis. So we have here BWF strings, or von Willebrand factor, that actually act as anchor points for platelets. So platelets start forming our blood clot and initiate the whole blood clotting cascade. So in normal hemostasis, we have regular BWF multimer regulation by ADAMTS13 enzyme. But in the case of TTP, we have unregulated BWF multimers. And so there's very low ADAMTS activity. ADAMTS activity less than 10% is indicative of TTP. And what happens is you get microvascular thrombosis and a different systemic mechanisms that occur, such as brain damage, kidney damage, and it really affects the small vasculature in the body. So both VWF and ADAMTS13 are multi-domain proteins. ADAMTS13 has the MDTCS proximal domains that are the catalytic activity components of that regulate the A2 domain at VWF by cleaving a specific tyrosine methionine bond within the domain. And in 2019, my advisor in his previous group did a phylogenetic analysis looking at vertebrate species, specific BWF and ADAMTS13, and then they designed phylogenic peptides where they were able to do functional analysis. And what they found is that all vertebrate plasma ADAMTS13 actually preferred the cattle VWF-A2 peptide to the human version, cleaving it from 1.5 to 15-fold faster depending on the species. And so this really laid the foundation for my project to determine sequence determinants of VWF cleavage by ADAMTS13 using cattle and human VWF-A2. So what I'm doing is we design novel wild type and chimeric VWF-A2, and we use ADAMTS variants, truncated variants, and allosterically unregulated variants to utilize this Alto SPR benchtop. So it is a very small, compact, new technology that allows us to do binding assays using this cartridge. And so here we use minimal sample volumes, 2 microliters, and we add our reagents, and we immobilize the first binding partner, known as your ligand, to the surface, and then we test analytes and probe them against to generate binding curves that kind of look like this. So this is a single cycle binding assay, and we're able to generate kinetic constants. And so specifically KD is the dissociation constant, which allows us to understand the binding affinities between protein-protein interactions. And with that, Pistol Pete tells you to check out poster number 32.
All right, I think I actually have a question. Finally, I can ask a question. So I was very curious, like this is all done in the in vitro, like are you also working with in vivo model, like a cell system or anything to test it out, these protein-protein interactions?
No, not yet, because we're really honing down on trying to determine the specific locations that where we're seeing this binding affinities change, and so with that, we're kind of doing the foundation biochemistry background, and then maybe looking to do it in cell models or animal models.
That's really cool. Thank you. So the next person will be the Cristina Flores. She's from all the way from California again, to Santa Cruz, UCSanta Cruz, and her title will be High-Throughput CRISPR screen identify the novel long non-coding RNAs that regulate the macrophage viability and function.
Hello everyone, and today I will give you brief overview about my poster that I will be presenting today. The title is Using High-Throughput CRISPR Screens to Identify Macrophage Viability Genes. So just as a brief introduction, macrophages play critical roles for infections and in the immune response. As I showed you here in this schematics, once the macrophage recognizes the pathogen, it elicits a response and the production of pro-inflammatory cytokines, interferons, and although in the past macrophages were not associated with adapted immune system, now we know that there's a crosstalk. So the activation of macrophages eventually leads to the activation of the adapted immune system. The end goal is the clearance of the pathogen. Because macrophages play a critical role in this immune response, we're interested in what controls their cell survival to understand more on the immune response. Now you can do one gene at a time or multiple genes, and we take advantage of CRISPR technologies. It allows us to do a whole genome approach, and that's what we do in our lab. Now, the most common CRISPR technology that all of us are very familiar with is the wild type or traditional CRISPR. This CRISPR system is used to target protein coding genes because they have a open reading frames. And what they do is that the Cas9 goes and creates a double strand in the open reading frame. And when there is a DNA repair mechanism through non-homologous end joining, the DNA sequence is shifted, and therefore the gene expression is disrupted. Now, we cannot take advantage of this technology for long non-coding RNAs because these long non-coding RNA genes, they don't code for proteins, and they don't have open reading frames. And that's why we take advantage of the modified CRISPR version, which is CRISPR-I. And so we use CRISPR-I, it targets the transcription start site, and instead of creating a double DNA strand break, it just creates transcriptional repression. And just very quick to give you an overview, this is what we did for our screen. We have the Cas9 THP expression cells. We have a high covering CRISPR library that encompasses for all the human non-encoding RNA genes that we know of. And then we collect samples at day 0 and day 21. And then we send up the samples for sequencing. And just to give you a little snapshot, this is the results that we have here. So in the first graph that I'm showing you is the 37 hits by the genes that I got from my screen. And then what you see here is basically the top 3 performing guides. And it was very surprising that the hits are divided in different types of long non-coding RNAs. So yes, we have different types. We have intergenic, those are genes that are found in between genes. We have intronics, and those are localized within the introns, and it was very fascinating stuff. From this screen, we have decided to focus on 2 of them. Long non-coding RF8 and non-malic because they neighbor protein coding genes that have been shown to be involved in viability. This is fascinating stuff, and I hope that you stop at my poster number 22 so we can continue this chat. And then lastly, I would like to give thanks to my mentor, Dr. Susan Carpenter, the IRACTA program at UCSC, and of course, Brown University for hosting us and organizing such a wonderful event. I'll take any questions now.
Any questions? Oh.
Hi, wonderful. How did you determine where the transcription start site is, where the promoter is, and how you can target that so effectively?
So we collaborate with University of California, San Francisco. Dr. Wiseman has organized a library, and he was the one that determines the transcription start size. Because they're poorly annotated, so they have generated a library of this transcription start size. And then to avoid false positives, we try different guides. So for each gene, we do 10 different guides.
Is Pol2 used for the long non-coding RNA?
I'm sorry?
Polymerase 2, RNA polymerase 2, is that?
Yes, it's used for the long non-coding RNAs. Great question.
Thank you so much. And then because of the time, we should move on to the last lightning talk.
Thank you.
So for this session, the last person going to be the Ryan Donnelly. He's from just here, University of Rhode Island. So he's also presenting the poster. His number will be 27. So again, if you want to continue the conversation with him, he's going to be right outside. And then after him, we have a great keynote speaker. She's waiting right here. So his title going to be the development of stably expressing Cas9 GFP, embryogenic cell lines of Sorghum for rapid analysis of guide RNA. Here we go.
So, hi, good morning, everyone. So yeah, I'm going to be talking about the development of the stably expressing Cas9 GFP embryogenic cell line of sorghum for the rapid analysis of guide RNAs. This was a project that was also very much driven by CMB 499 students from our school. So sorghum, you might not be familiar with, is the 5th largest cereal crop globally. These are blurry pictures taken from our greenhouse, and these are immature embryos, which I'll explain a little bit more in a moment. But so this is an important food source for several parts of Africa and Asia, and it is also a crop of interest for the DOE as a potential biofuel. So what we do in our lab is we do traditional agrobacterial transformation. And we do this on those immature embryos. So you may have noticed they're very small. We actually extract the embryos from those little seeds. So you know the meme of surgery on a grape? That is what we do. This is very labor intensive, very time consuming. It can take about a full year before you're going to get a fully regenerated plant. And quite frankly, you're not going to get a whole lot of them for how many embryos you actually go through. So we have the goal of ultimately getting to a tissue culture-free genome editing system. This is a very ambitious project, but in the very first step of all of this is we want to actually start using CRISPR-Cas9 for our transformations. So to achieve this, we've actually developed a cell line called PNG111, and it is expressing Cas9 and GFP off of the same ubiquitin promoter. This is great for us because we can now use GFP effectively as a visual marker for where we have Cas9 expression. This also doubles now as being a visual target for Cas9. When we supply guides against GFP, we would expect to see GFP turn off, and that's a good visual marker before we get to the sequencing level. Now, for delivering these guides, we've actually been using something called single-walled nanotubes, or SWINTs. And these can carry either DNA or RNA on the outside of them as their cargo. And so for us, obviously, that is guide RNAs, and we are testing this against the GFP. We had, I think, 3 guides designed. We've been actually testing 2 of them. And another thing with the PNG111 is that it's actually able to exist as a stable callus line. So we just continue to subculture it, and this is what allows us to test this more rapidly because we don't have to go through that whole year process. We can actually just deliver these guides directly to the callus, and within about 2 weeks, the half-life of GFP, we should actually start to see a decrease in GFP. Then we can get that sequence data. And what's also really great about this system is it's not just to test the guide RNAs before we go into the more laborious intensive process. We can actually test different guide delivery methods. We've been using the SWINTs, but some of our collaborators are currently using viral vectors and also cell-penetrating peptides. And this is what the callus line actually looks like. Just because, again, this is probably something not a lot of you are familiar with, so just to give I'm a very visual person, so I'm kind of getting a picture of what people are talking about. And I want to say, thank you to everyone. Thank you to all of our members of our lab, our collaborators, particularly the Marquita Landry and Daniel Roxbury. And and I hope you all come see Chibuki and myself at poster 27 for any further questions. Thank you.
Any questions for Ryan? I have a question. So, to be honest, I never seen any model system like Sorghum, and so you told me that the picture we saw was embryo. So,
that's a panicle, and those are just immature seeds. Then the embryo is inside those seeds, so we actually have to go through a whole surface sterilization step, and then we actually have to extract those embryos, and they're very delicate too. So again, it's also a very technical skill. I know I really labored into that. It's labor intensive and time consuming, but it's also a very delicate task also.
so I imagine we also know about the genetic genomics of this sorghum too, right? For the
We do
CRISPR
know a
Cas9.
little bit, so it's not a model system, so it's not quite as well studied as some other systems as we would like. Like a lot of things actually end up being done in like Arabidopsis and Soteria, I believe, are other more model systems people tend to work in first. We actually had a little genome mishap in our lab. We thought we had a VTX430 for the longest time, and then at some point someone mislabeled something, and we've been working in the wrong genome for some time. We finally got that fixed, and magically our transformation numbers have improved a lot. So, also, as just a little note of that, labeling really matters, everyone. Please label your stuff accurately.
I'm trying to teach myself in my undergraduate, but it's not easy. But yeah, very interesting. Thank you so much.
Thank you.